Our next speaker is Professor Gabriel Gwanmesia from Delaware State University. His talk is about study of the thermo-elastic properties of hydrated wadsleyite by ultrasonic interferometry with synchrotron X radiation.
All right, first of all, I'd like to thank the coordinators and organizers of the workshop for the opportunity to present our work. This is ongoing work on the measurement of the thermoelastic properties of hydrated wadsleyite. I think from the previous speaker, we see the importance of understanding how water affects the elastic properties of materials. Many collaborators with different expertise that I acknowledge at the end of the presentation are involved in this study, from the sample preparation, the detailed characterization of the material, and the elasticity measurements. I want to first speak about the summary of what we've done so far with regards to the elasticity measurements, which clearly shows the importance of synchrotron radiation in the success of the work. Then, as requested by the workshop organizers, I will talk briefly about our efforts in broadening the participation of minority students in the geosciences because of the persistent poor minority representation in the field. In fact, statistics shows that the geosciences ranks only higher than physics at the bottom of the STEM fields in minority representation. As I said before, we already heard from the previous speaker about the importance of understanding the properties, the effect of water on the elasticity of materials. So many studies show that they affect other properties, electrical properties, thermal conductivity, rheological properties, and of course, the elasticity of the materials are affected by water incorporation in the mantle minerals. So far, there are not many measurements of the high pressure and high temperature elasticity, and in particular, the temperature dependence of the elastic moduli of the hydrated phases. Many temperature derivative data source, or the main one comes from compression studies, which yields only information about the isothermal bulk modulus, but not the shear modulus. There are, of course, some Brion scattering measurements of the bulk modulus of the pressure derivative of the elasticity of hydrous wadsleyite and hydrous ringwoodite, and also a few temperature derivative study, but still only mainly on the hydrous ringwoodite. So using the ultrasonic techniques and the ability to make suitable polycrystalline sample, we are in the process of conducting elasticity measurements at simultaneous high pressure and temperature for hydrated wadsleyite phase, and hope that if we can clear and do it well, and then we move on to study the hydrous ringwoodite. So this is the cell assembly that we developed for hot pressing materials for ultrasonic specimens. The critical component of the cell assembly, this is actually a compressed standard cell. The critical modification that we made to the cell was to replace the MgO insulating sleeve around the sample by sodium chloride. And during the development of the cell, we actually showed that when you hot press material inside an MgO sleeve, it took on the hourglass shape and easily cracked on decompression compared with cylindrical and crack-free specimens hot pressed in the sodium chloride sleeve. So for this hot pressing the wadsleyite sample, the starting material is phosphorite, with brucite to introduce water into the sample, and then charge balance by adding silica. When we started with a mixture of MgO, silica, and brucite, we end up with mixed phases because the phases are phase E and others are very sensitive to temperature. So this was the best way to proceed. So we also use gold palladium capsule to minimize water loss, but sometimes we just use platinum capsule because it's easy to extract the sample, hot press inside a platinum because it's relatively soft. The PT conditions are shown here. The PT condition is 15.5 GPa and 1000 degrees. The only thing that we vary is the annealing time, which depends on the water content of the sample. This is actually in contrast to the dry sample. It takes only 1 hour and the sample is completely annealed and good. So we do extensive characterization on test specimens to optimize the condition of fabricating suitable samples for ultrasonic experiments. There are very stringent requirements for the sample in order to be suitable for ultrasonic measurements. We conduct X-ray analysis to confirm the sample is single phase. We measure the bulk density and compare them with X-ray density, and also measure the water content in case of the hydrated sample, which is something I'll talk about later. So these are scanning electron microscopy, microscopy analysis of the polished and cracked surface of wadsleyite containing 0.73 weight% water, which we already published. The scale bar is 1 micron on the polished surface and 2 microns on the cracked surface. As seen here, there is no observable porosity at the triple junction of the polished sample. The average grain size estimated from the cracked surface is 5 microns, and the orientation of the grains is fairly random. Fine grain is essential because the velocity measurements involves the propagation of high frequency, usually 20 to 70 megahertz signal through the sample. So as shown at the bottom of the screen, the bulk density of the sample that we are talking about and the X-ray density based on the relationship here from Mao et al. Agree excellently. We also show here the X-ray, and here we show the perfect match between the X-ray diffraction pattern and the unit cell volume taken before and after the high pressure and high temperature ultrasonic experiments, showing that the integrity of the sample is actually maintained during the experiment. Also, on the right side we show that there is no water loss from the sample as shown by the FTR spectra of the sample before and after the high pressure experiment. This is a schematic diagram of the setup for the equation of state and elasticity measurements at the 6 BMB beam line at the APS. The pressure is generated by the die-type cubic anvil apparatus interface with the X-ray that enters and exits the sample chamber through the anvil gaps. Attached to the bottom part of the press, one of the anvils is a dual mode lithium niobate transducer for generating P and S wave into the sample and receiving the reflected signal. The station is also equipped with a CCD camera to capture the sample image during the experiment for determining the image length for comparison with the length obtained from the unit cell volume. So at the right side of the right lower diagram, there is the magnification of the cubic cell assembly shown that we will discuss in the next slide. So the top right is a schematic diagram of 4 of the 6 anvils of the dia with a dual mode piezoelectric transducer mounted at the back for the lower anvil. The cell assembly on the right, top right, is made of boron epoxy, and the resistant furnace heater is a graphite. A double-phase polished alumina rod transmits the acoustic signal into the sample, which is inside a boron nitride sleeve and supported at the far end by sodium chloride, sodium and boron nitride mixture. So accurate temperature measurement is achieved in this cell by direct contact of the tungsten rhenium thermocouple on the sample, as shown here. And in some experiments, we actually, to create symmetry, we have mounted two thermocouples, and we can read the temperatures. The temperatures match within 2 to 3 degrees centigrade. We also insert a 2-micron gold foil at the top of the sample and at the bottom for imaging the sample length. The precision for direct measurement of the sample length using this technique, that's using the image, is about 0.2 to 0.4%. The top left hand is a schematic diagram of the interferometry technique. The signal from the dual mode transducer actually mounted at the back of the anvil. It will travel through the buffer rod. At the buffer rod sample interface boundary, part of the wave is reflected due to difference in the acoustic impedances of the adjoining materials, and part of the wave is transmitted through the sample. The transmitted signal also reflects at the far end of the sample and returns to the transducer. The top right diagram is an example of the input signal and the reflected echoes from the buffer rod B1 and S1, the echo S1 that has made one round trip in the sample. So we determine the P wave, P and S wave travel time by a pulse echo overlap technique. That is by matching or overlapping the two echoes from the buffer rod and the sample cycle for cycle, as shown in the lower bottom part. The precision of this travel time measurement is extremely good, about 10 to the minus 3 to 10 to the minus 4. This diagram here shows the PT path that we've used for studying the hydrated wadsleyite at high pressure and high temperature. This is different from the conventional path used in most previous experiments, where we first compress the sample to maximum pressure. After heating the sample to high temperature, the cell relaxes and the pressure in the cell drops, usually about 1 to 3 GPa, depending on the cell assembly and the temperature. The data is then collected during the cooling cycle. The pressure is decreased and the sample is reheated, and the data is again collected during the cooling cycle. But the maximum temperature for the hydrous wadsleyite measurements is only 300 degrees centigrade, about 600 K, to avoid dehydration of the sample. So we follow a unique PT path. We first increase the pressure moderately, heat the sample to 600 K, and then take data every 100 K on cooling. We generally wait at each point for about 3 seconds to equilibrate the temperature. So back at room temperature, we again increase the pressure. We reheat the sample to 600 K and repeat the data collection process. In all the experiments that we've done, we also try to collect a few data points on the way back, that is on decompression. This is shown at the bottom of the the bottom figure actually shows a plot of the travel time of P and S wave for the sample, for the data collected at the end of each of the cooling cycle. Compression and decompression travel time shown in red and blue respectively, they are consistent for both P and S wave, and both data sets, both on compression and decompression, decrease systematically with pressure. We increase the precision of determining the pressure dependence of the acoustic wave velocity by having this many data points in the analysis. Again, this is in this diagram, I showed the plot of the P and S wave. The P and S wave, as a function of pressure, you see that the travel times decrease linearly and systematically with pressure, and also the length of the sample shown on the lower left corner. For this is also the case for the P wave and S wave velocity plotted as a function of pressure at the top in the top two diagrams, and for K and G plotted at the lower in the lower diagrams. We see that in all cases, the velocity and the moduli, they all increase linearly and systematically with pressure. We have carried out a measurement on a number of samples, several different samples, maybe 4 or 5 sample compositions have been measured, and we see this behavior in all the samples. Here we summarize the data for the sample that contain 0.7 weight%, for the Wadsleyite sample that contain 0.7 weight% water. The data in red is a comparison of the results that we obtained from linear fit of the entire data set. The second data in red is from finite strain analysis of the entire data set, and the last set of data comes from fitting the linear regression of those set of data that we obtain at the end of the cooling cycle. As seen in the table, all K and G and their variation with pressure from the 3 analysis methods are in excellent agreement, except of course for the pressure derivative of the shear modulus from the finite strain analysis, which falls out of the mutual uncertainty of the data. We also measured the anhydrous phase, also shown here in red, the one with 0% water. We did that in order to validate this technique, because most of the time when we measure the elasticity of the anhydrous phase, we usually measure it at very high temperature. So we wanted to repeat the same experiment here to validate the results. And you can see that the K and G from our data for the anhydrous phase is in good agreement with the results from Brion scattering and other ultrasonic experiment study from previous studies in the table. But although the pressure dependence of our K in this study for the anhydrous Wadsleyite are internally consistent, we can see that they are high compared to the Brion scattering data. Some ultrasonic data measurements show the similar discrepancy that could be caused by the residual stress in the boron nitride ultrasonic cell assembly, and especially in this case when the temperature is very low for measuring the Wadsleyite samples. We have recently modified the cell assembly during the pandemic. That's what we did, by completely surrounding the sample with sodium chloride to minimize the stress on the sample. And we just await the opportunity to test out the cell assembly during the upcoming, when we have the opportunity to carry out new experiments at the APS. Lastly, we can see that the reduction in the bulk and the shear moduli due to hydrogenation of the Wadsleyite from this study is compatible with the Brion scattering study shown here for Wadsleyite containing 0.0 weight% water. So recently, we just completed analysis of the water content following 2 different techniques. So we show here in this diagram, in this table, that the water content was measured using the SIM method and also by the microanalysis technique. But we see from the table that the EPMA technique shows small water, the water content is consistently smaller than measured in the by the SIM technique. And we associate that the difference to the effect of grain boundary on the SIM technique, where the grain size, where the beam size is about 25 microns compared to 1 micron for the EPM technique. So we are going to constantly use the water content measured by the EPMA technique for further analysis and discussion. In this figure, the top right hand, we plot the bulk and shear modulus as a function of water content at the top right hand corner. The three blue ones are symbols are data from 3 hydrated samples that we have measured. So different water contents that we've already measured at the APS. We have added to that the wadsleyite results from the sample with 0.73 weight%, where the water content was actually measured by the FTR technique. And also we add to it the data for the anhydrous phase measured from the same study for comparison. As shown in the top right-hand corner, we see that when we add, we observe first a significant drop in K and G of about 3.8% and 3.7% respectively by just adding 0.3 weight% of water. So the first drop is from adding 0.3 weight% water in the wadsleyite. And then as we further add water into the sample, we find that the effect is not so dramatic, it's almost linear. That means that once we add up to maybe that 0.3 weight%, any further addition of water does not seem to have significant effect on the bulk and the shear modulus. And at the bottom, we showed the bottom right-hand corner, we have plotted the pressure derivative of the bulk modulus and the shear modulus as a function of the water content in the lower right-hand corner. And we see that both K and G are almost independent of water content. And this has already been shown by some studies by Brion scattering. So water does not affect K prime and G prime, but causes a drop in the bulk modulus and the shear modulus. Here we plot the temperature derivative of the bulk modulus and the shear modulus as a function of the water content. We're looking for this pattern, and you can see that the first 2 data is from the anhydrous wadsleyite that we measured. Then the second one is for the 0.3 weight% water, and then subsequently other water content. Again, the brown symbols are for the hydrated phase, and we can see that it shows that when we add temperature, the temperature actually affects the bulk modulus, the bulk modulus more than it affects the shear modulus. You can see from the data. This is unusual because most other measurements that we've seen, especially for the anhydrous sample, whether with ion or without ion, they show that the shear modulus is always higher than the effect on the temperature on the shear modulus is higher than the bulk modulus. But for the water content for all the wadsleyite samples that we measured, it is showing that it affects the shear modulus, the bulk modulus more than it affects the shear modulus. So we are still going through, we just obtained the water content, and we'll spend some time now looking, first of all, clarifying the issue about the higher pressure derivative of the bulk modulus compared to the data from the Brion scattering, and also see whether by changing the cell assembly, anything will happen, whether there will be some changes in the temperature derivative of the elastic properties too, because these are the first measurements and we don't know what will happen, whether there's any effect of stress on that. So we have to clarify this and then go on to do the geophysical implications.
1 minute.
Yeah, so in summary, we've seen that we can now have high temperature, high pressure elasticity data of mantle minerals now that we can make samples by ultrasonic interferometry technique. We also showed that 0.3 weight% of hydration of the wadsleyite decreases K by 3.8% and 7.8%. And further increase in the water content does not seem to have any significant effect on K and G. We show that the pressure derivative of the bulk modulus so far is independent of water content, and that we can resolve the issue of K, pressure derivative, by actually modifying the cell, introducing sodium chloride. So that's it for this part. As I said, the coordinators of the session asked us to talk a little bit about the educational initiative to increase diversity in the geosciences. So in 2011, Bob Lieberman led a proposal to the National Science Foundation under the opportunity for enhancing diversity in geosciences. We titled the program Career Path for African American Students from HBCUs to National Lab, and with Lars M. and myself and me as co-PIs in the proposal. Initially, we obtained about 200K from the NSF, but leverage and further support came from the Fulton Source Science at Brookhaven, the Graduate School at Stony Brook, and the Bridge to Doctoral program at Stony Brook. So by the end of the program, from 2011 to 2016 or 17, we had already garnered about 711K. So the aim of the Career Path program was to recruit underrepresented minority from science and engineering. And from HBCUs, mainly my school was going to be the main target, of course, and then to expand into other HBCUs into the Stony Brook Geoscience program. The students took formal courses at Stony Brook and conducted research projects leading to an MS degree in geoscience instrumentation. So the idea also was to develop marketable skills in this, the trainees in the emerging science of, in the emerging field between science and technology, so that they could go on to gain employment in the national, in any of the national labs with knowledge of both science and instrumentation. The money helped pay the tuition, stipends, and health benefit of the students. The students carried out projects at Stony Brook and also at the National Synchrotron Light Source. All these projects, they were supervised by Lars, and we greatly appreciate his effort in carrying on that responsibility. So here are the graduates, the product of the training. Ashley Thompson and Adair were both students. They got their BS degree in physics from Delaware State University and went on into the program. Melissa Sims was a geophysicist from the University of South Carolina, who was also entered the program. She finished the program and then went on to obtain her PhD degree in 2008. Sorry. Brandon came in through a very unique process. He was an intern at the Brookhaven National Lab in one of the summer internship program and worked under myself, under Lars and me for 2 years. We recommended him into the program. He came, joined the program, obtained his master's, and then went back to the Virgin Islands to teach in the university. So when the program came to an end, the NSF program, we applied for funding through Compress and obtained program for 2 years. We changed the title of the, or the level of the engagement to Career Path for Underrepresented Minorities from MSI, not only minority Black institutions, but to minority serving institutions, to National Lab. So minority in this case included African Americans, Hispanic Americans, Native Americans, and students from socio-economically disadvantaged backgrounds, in which we were able to hire Kathleen LaFranca, who came in, did a project with Lars as usual, and then obtained her master's degree in 2019. All students gave presentation, poster presentation at Compress meetings, AGU meetings, and various seminars. And then we also published this, the results in the EOS, this EOS journal. Finally, I want to talk about the students that we have at Del State. You know that even at undergraduate levels, they are underrepresented. So through RE programs and funding from the NSF, we usually obtain money to support students, give them stipends to work, to do research with us so that we can train them and encourage them to go on to graduate program. These are the students. I show these slides because they were working at Brookhaven at X17B2 beamline. So Richard was a student here. He's now a graduate student at Stony Brook. And they went through the career program and graduated. These are other students. This is Li Ping Wang, giving them training before starting experiments. And they were all students that I brought from Del State to train them under NSF support. So finally, I just want to thank and appreciate all my colleagues and collaborators. A lot of work has to go in just looking at the sample and making sure that the sample is of good ultrasonic, for good ultrasonic studies. So I have many collaborators with different expertise that took part in this experiment. And of course, the support for my work comes from NSF, compressed with the cell assembly, and DOE, APS, DOE, Office of Science, for giving me the opportunity to carry out the experiments at the APS. Thank you.
Thank you very much, Gabriel, for the nice talk. And it's always nice to see like a student's smiling faces.
Thank you.
I have we have time for one quick question from Subor, please.
Oh, thank you. Thank you. That's a very exciting result. So, Gabriel, I just have a very quick questions. When you characterize the water content, I recognize you have two technology, EPMA and the FTIR. Does the FTIR have some influence on the grain boundary effect of those water content?
Okay, that's why we have not yet, we just finished this, we just got the data for this analysis recently. And I just, in my talk, I just used the data to see where they fit in the data. It will be useful for us to do the FTR on all the samples and see how we can compare them with the results of from this water measurement, because we only did it on one sample, so it's difficult to talk about the effect of grain boundary on the measurement.
Okay, thank you. Thank you.
Thank you very much. So speakers can continue replying to questions in the chat.